Okay, let's see if this works. There we go. So that I would actually dedicate this lecture to my mentor, Audrey Holland. I hope Audrey has logged in, and if she's watching, then I think it's appropriate to say that Audrey, for those of you who don't know me, Audrey was my PhD mentor at the University of Arizona. And although she was an excellent mentor, she has been even better as my mentor post-PhD study, in that she has been somebody who I've been able to bounce off ideas, to ask to do grant reviews, manuscript reviews, and she's been my rock through a lot of different things. So Audrey, this one is for you. Although I will say, once you've heard this lecture, you may want to disown me, but we'll see. So I did spend some time preparing for this talk, talking to Audrey on a recent trip to Tucson, Arizona, where I stayed with Audrey. And I decided that during that time that I was going to record our conversation. I have about an hour and a half of recordings. What Audrey has to say about aphasia, I think, is very important. She's certainly one of the leaders and giants in our field. And what I decided to do is not just keep that audio recording for myself, but rather, with Audrey's permission, we are going to post the audio from that conversation on the Aphasia Lab podcast. That's a page that we're going to be having up and going probably I would say in the next 3 to 4 weeks. So if you're interested in hearing what Audrey had to say and some of the things that we agreed and disagreed on, I would encourage you to listen to that podcast. So regarding this lecture series, which we call Aphasia United, the first lecture was given by Dr. Linda Worrell, who is a professor emerita at the University of Queensland in Australia. And the topic of Linda's lecture was on the social cure, and it's already available on the C-STAR website. I am today giving the second lecture, which focuses on the neurobiological cure. And the next lecture will be by Dr. Miranda Rose on the behavioral cure. And it will be hosted by collaborations of aphasia trialists on May 6th of this year. And then we will have a final lecture, which will be given by Dr. Miriam Brady, and her title is Finding the Cure, and it will be hosted by the Aphasia Center for Research Excellence. I don't have the date for Miriam's lecture yet, but I'm hoping that we can announce that soon. So like I said, my partner in crime here is Dr. Linda Worrell, and she has really been the driving force behind Aphasia United and our lecture series on curing aphasia. As Linda explained in her lecture, this really came about as a result of a Twitter conversation that she and I had regarding progress in our field and lack of overlap between the different approaches in aphasia research. And we agreed that starting this lecture series might be a good start to fostering collaboration. And the question that I posed we tackle in our lectures was, if we ever manage to find a cure for aphasia, what might that cure look like? Like I said before, this is very different than from my customary lectures. I've taken a lot of liberties in my thinking about the possible future and what that future might entail. And as you will see, you may think it's completely unreasonable or unrealistic. I'm fine with that. This is not meant to chart the course forward. Rather, what I was thinking about here was representing a single course forward that I thought might be realistic, or at least something that was worth dreaming about and then pursuing. So going back to my online conversation with Linda, we agreed that we have become very siloed with regards to our approaches to aphasia treatment, whether it's in the domain of functional versus impairment-based approaches, or with regards to neurobiological versus cognitive versus linguistic approaches. And there's also another dimension there where some separation may have occurred across live participation approaches versus language improvement approaches. So we suggest that it might be useful to take a step back and try to integrate different approaches to aphasia research and aphasia therapy particularly, and thereby improve the crosstalk and the quality of our work. Looking at where when I got into this field, I was starting to work with Audrey in the late 90s, so 97, 98. I would suggest that the overall progress in improving recovery in aphasia has in some ways been incremental, and I suspect some people might suggest has been almost non-existent. I spoke to Audrey about this when I visited her, and I posed the question to her whether in the USA, for example, persons with aphasia are receiving better therapy today than what somebody was getting, let's say, 40 years ago. And Audrey agreed with me that that it may not be the case, that 40 years ago, we were doing therapy that was subpar compared to what we are doing right now, or the management and the approaches that we're using, but they are better and certainly not reaching the number of people that we need to reach to make a big difference. So in my opinion, the status quo is unacceptable. I think that persons with aphasia deserve more decisive and bolder action to try to improve their recovery. And what I will discuss today is just one of the many paths that I think we might want to consider. And I want to emphasize that the solutions that we seek will not be found simply within my field of expertise, which is the neurobiology of aphasia recovery, but I really think that it will be integral, but at least I think neurobiology and neuroscience will be a part of the solution moving forward. So curing aphasia, I'm going to focus on four main things today. The first one, I'm going to talk about what I mean by curing aphasia, because I realize that it might mean different things to different people. And then I will talk about why now is a good time to do so, what it will take, and what is a reasonable timeline. And I will pretty much end with that. I want to emphasize yet again that there is lots of speculation included here, and that many of the steps that we will need to overcome will not be easy, and neither are they straightforward at this time. So I think it's best to start with a definition. I'll go through this very quickly. I realize that most of the people listening to this already know this. I hope that we are reaching some students at least today. So some of the things that I will intersperse here with my lecture are really meant towards to target students and hopefully new investigators in our field. So when we talk about aphasia, so it's we all know this definition. It's a language impairment caused by brain damage that results in communication difficulties. It certainly affects the individual and communication partners. It induces social handicap, and in most cases is life-altering. Now, what Linda talked about is really these four bottom items. What I'm going to be focusing on mostly is language impairment caused by brain damage. Not saying that these other things are not important. It's just because of the focus of my talk is such today that I'm going to focus on the first part and not so much the others. So when I talk about curing aphasia, I'm talking about restoring or enhancing residual brain function so it can support complete restoration of language functioning. So when you look at the top multicolored image here, what you see at the top here, it highlights the main speech and language area of the brain. It is not surprising that the brain images that you see scrolling at the bottom, and almost all of those brains, they showed an overlap between the damage, which is what you see in the darker shade on that image, and the language areas of the brain. Now, what we are talking about today is how do we literally overcome the effect of the lesion on the brain and help the brain recover so that it can support or restore language function, and then as a result, improve communication status. I realize that the mere suggestion that we will someday cure aphasia may seem like mere hubris, but I would suggest that progress in the field of neuroscience in the past decades does give some glimpses of what an eventual cure might look like. So curing aphasia, why now? I would say that even if the solution to the problem does not seem within reach, at least working towards that solution could at least partially ameliorate the problem. So even if we don't see what the end is, I think we need to work towards whatever we think that solution might be, and in doing so, we might make progress that would benefit folks with aphasia. We understand better than ever that the effect of aphasia on the quality of life, and like I said before, in my opinion, the status quo is not acceptable. In the USA, funding for aphasia treatment research is available. I think we need to be bold and go for the big grants to fund projects that will make a difference for treating aphasia. I understand this is very loaded. There's a lot of things that are included here, but I really feel like, and I'll get to this a little bit more later, that the major progress that is going to be made needs to be done with both the combination of smaller projects, but we cannot ignore the larger research questions and the larger research projects that need to be carried out. I would say that our field, and now I'm talking about treatment of aphasia, unfortunately, we've had a lot of aphasia treatment studies. If you just look at a review of the literature, there's been a lot of aphasia treatment studies done. Even going as back far as 1865, Broca was already talking about potential aphasia therapy. But in spite of all that, in spite of all those studies, we still are probably not, we haven't come as far as what most of us would like. And I think one of the reasons for that is because a lot of our studies are very small. And I would suggest most of them are vastly underpowered. And I don't think our field is that much different than many other fields. If you read this paper that I highlighted here by Ioannidis from 2005 in +1, the title of his paper was Why Most Published Research Findings Are False. One of the things that he really hammers home, which I think applies to our field, that small studies are very likely to have the incorrect result. And therefore, if you always focus on these underpowered studies and never move on to the larger research projects, we are in danger of all of our studies, well, most of our studies, not having much effect on clinical practice, and I would argue very limited generalizability. Incidentally, for any students out there, I would think this would be required reading, this paper by Ioannidis. It is the most downloaded paper ever on the Web of Science, and it's available free of charge. So curing aphasia, again, going back to why now. So the first step in tackling a problem is to describe the problem in detail. Another step is to look at where the field stands today. So I am going to spend just a couple of slides talking about progress in understanding normal speech and language processing in the brain, and then very briefly pivot to understanding cortical organization associated with aphasia recovery. So one of the studies that I really like that came out fairly recently, this is by Shane et al. This is F. Fedorenko's group at MIT, showing that language organization in the brain, with regard, language very much relies on language-specific areas, especially in the left hemisphere. There might be some influence from multimodality areas or other regions of the brain in language processing, but by and large, the areas of the cortex that are important for language get very little or minimal help from other systems in processing language. And I think that is important when I start talking about how I think the brain supports recovery from aphasia. Again, so why is this important for us? I would say, based on the literature so far, reorganization that supports aphasia recovery, I think, is mostly confined to residual language areas in the brain. And so the reason why individuals with large lesions or extensive damage don't recover is not only because they have less neural tissue remaining, but also because the areas where most of the positive brain plasticity can occur was already destroyed. Now, going to the cortical reorganization associated with recovery from aphasia, I don't have a lot of time to spend extensive amount of effort on reviewing that whole literature, but I just want to highlight this one study that was recently published, I think in the last couple of weeks, in the journal Brain by Stockert and colleagues. This is a group led by Dorothea Sauer in Germany, and they were looking at brain changes in early aphasia recovery. So using functional MRI to look at language activation in the acute care, again in the subacute care, and then finally in the chronic phase. And what they did was that they compared those 3 different phases across 32 participants, showing that the changes in brain activation really depend on where in the brain the damage occurred. So they looked at 2 groups. First, I want to show here's brain activation in normal individuals. This is associated with normal language processing. And then they had 2 groups, one group that primarily had damage to the temporal parietal lobe, and then another group that had damage to the frontal lobe. And what you can see here is progression from acute to subacute to the chronic phase. And the changes in activation really depend on where the damage occurred in the brain. That is going to be important when I start talking about some of the things that I see in the future as being a potential solution for curing aphasia, in that individual differences really matter. So I doubt that we're going to find some kind of a neurobiological approach to treat aphasia that will be applied to all persons with aphasia. So I think individual differences matter, and I will also talk about that when I get to talking about aphasia therapy. One of the main take-home messages from the Stockert et al. Paper, and I'll just read this out here, was that overall, our analyses suggest that language recovery takes place in a pre-existing network and does not strongly rely on the recruitment of additional brain regions. This implies that for highly specialized functions such as language, restoration within left lateralized language and compensation in domain general networks dominates over de novo recruitment of functionally unrelated networks. Now, I really would say that in my reading of this paper, I would bet that most of the recovery that they're seeing is really related to changes in brain activity in the residual brain, in the residual language network, rather than in domain general areas. But that's a talk for another day. The other thing that I liked about this study, which goes very much along with what I think about how aphasia recovery is supported in the brain, is that they found changes in perilesional activity was associated with improvement in aphasia. So when I say perilesional, you can think about the damage in the brain. So those regions that are closest to those regions that are closest to the lesion, that's where much of the brain plasticity occurs that drives aphasia recovery. Now, that's a little bit loaded, and there's a lot to say about that. But in general, it seems to be the case that for good recovery to happen, regions that were already doing language processing before the stroke really need to sort of pick up the slack and make up for the language regions that were damaged in the actual stroke. So what are some viable mechanisms? There are many different ways that you can look at it. At the cellular level, you can certainly talk about axonal sprouting, dendritic branching, neurogenesis, remyelinization of axons, alterations of excitability, and stem cell implantation. I really think that there has been some progress in stem cell implantation. It's different than the other processes that I mentioned there, in that it's an outside process that you induce upon the brain. But I would, in my opinion, the progress that we maybe looked towards 10 to 15 years ago with regards to stem cell implantation, as far as I can tell, and I might be completely wrong, there could be something that I missed in the literature, but it has not shown the kind of improvement that we were hoping for. What I am going to talk about today, however, is I'm getting a little bit of feedback right now. You know why? Anyway, at the network level, I'm really talking about recruitment of residual language cortex, recruitment of domain general networks, which I think, again, that's a possible recovery mechanisms, although I would not put my money on it. And then recruitment of homotopy regions in the right hemisphere. I think that right hemisphere recruitment happens. I think it mainly supports recovery in individuals with very large left hemisphere lesions. However, as we all know, folks with very large lesions and initially very severe aphasia tend to show relatively minimal recovery. That doesn't mean that everybody who has a large lesion is not going to recover. And every once in a while, we see somebody who makes major gains. But everything else being equal, I would bet that the folks with the best outcome are the folks with some spared language cortex following the damage. So how do we improve recovery from aphasia today? We primarily look towards aphasia therapy, albeit with different kinds of approaches to improve recovery from aphasia. Other approaches that have been explored are drug therapy and electric brain stimulation, which I will review here very briefly. So behavioral therapy, I'm a firm believer in aphasia therapy of every different kind. I think that there are many different approaches that have been suggested. And one of my favorite papers on aphasia therapy is this one by Katharina Breitenstein and a large group of investigators in Germany. This was a randomized controlled trial. It was a phase 3 trial where they compared impairment-based aphasia therapy to no therapy, and the folks who were allocated to the no therapy group actually got delayed therapy. So everybody who got enrolled in the trial got therapy, and they looked at changes in both speech production on the adult, and they also looked at changes in communicative quality of life on the sac wall. And what they found, which is very important, was that there was a statistically significant improvement in the intervention group, so the experimental group. However, there was no improvement in the control group in the speech production measure on the adult. This was a medium effect size, so a Cohen's D was a medium effect size. And as importantly, and many people would argue more importantly, they found a statistically significant improvement on the sac wall. And although that effect size was a small effect size with regards to Cohen D. But I think very important study, certainly for students, if you're getting interested in aphasia therapy, this is one of the studies that you may want to start with. Now, I remember at a conference a couple of years ago, I did ask the senior author, Annette Baumgartner, whether they saw an improvement in all their participants. So even though they saw a group improvement, did they see an improvement in all the participants who were treated? And her response at that time was that about 40% of their participants showed either no improvement or actually a little bit of decline. And I think this is very similar to what we are finding here. So that means that most of the effect is really being driven by a subset of the participants who get involved in the studies, that they really drive the overall effect. And I'll talk about that in a little bit. I'm going to show you something that is from one of our studies here at C-STAR. We have a treatment study where we enroll participants with chronic aphasia. They undergo phonologically focused therapy for 3 weeks. So it's 15 sessions of therapy, and then they take a break, and then they come in for semantically focused therapy. It's a crossover design where half of the participants start with phonological, and the half of the participants start with a semantic treatment. And then we have looked at just overall improvement, comparing baseline to 6 months post. That's something that 2 people in my lab have been looking at, Dr. Alexandra Basilakos, who is a research assistant professor in my lab, and Julius Fridriksson, who is a Ph.D. student with me. Looking at just improvement in naming, if we rank the folks who whose data we've looked at so far, that's 59 participants. I think we're actually up to about 75 for the participants who've completed all aspects of the study. If you rank them from the least improvement here on the left to the greatest improvement on the right, what you will see, and this here on the y-axis, let me see, on the y-axis is actually change in correct naming, and these are just the change for the different participants comparing baseline to immediately post-therapy. There are actually a couple of participants who really went down, got a lot worse. This is proportional improvement, so room for improvement compared to actual improvement. So what you will find is that there's a lot of variability across the subjects, and this will become important when I start talking about what I think is a solution or one potential solution to curing aphasia. If we look at everybody here on the right, we could probably say that those participants showed some demonstrable improvement in the outcome. And as a side note, we're also looking at discourse. We're not just looking at naming, we're looking at discourse measures. And I think that at least looking at our preliminary data, the story here is very much the same. There is a subset of the participants who really responds well. There's a subset of the participants that shows absolutely no improvement or maybe just getting a little bit worse. And then there's another group here in the middle that might be showing some response, but not much. So when we talk about behavioral therapy for aphasia, I really think that individual approaches are extremely important. However, I do think that those individual approaches need to be driven not just by our experience as clinicians, but also need to be driven by empirical science. It is our responsibility as the scientists, as the researchers, to get those data out among the clinicians so it can benefit the actual participants that need it. So the other thing that I want to say really quickly, that if you compare the improvement with phonological therapy, which is what we have ranked there with the blue columns, the changes in naming, correct naming with the blue columns, and changes in correct naming with the semantic therapy, what you will see is that there's a huge variance. There are some folks that only respond to one kind of a therapy approach, like this person right here, only improved with the semantic therapy. Some folks that improve with both, at least like this person right here, a couple of other folks here at the other tail. And then there are folks here, many folks that actually did worse following the phonological therapy, but a lot better with the semantic therapy. Now, I don't want to steal Alex and Sifus's thunder because they are working on papers describing this in our larger sample. But what is interesting about this, and just as important, that some of the factors at baseline that predict outcome with semantic therapy are factors that don't predict outcome of phonological therapy, and vice versa. Then we have other factors that, for some participants, if they scored high on a given test, they responded to one therapy approach, but if they scored low on the same test, they responded well on the other. So a lot of data here that we are going to be sharing here in the not too distant future, but again, look for those papers from Alex and Sifu's coming out hopefully sometime soon. The other two other areas that I want to touch on really quickly, drug therapy. I'm not going to go into much detail here. There are some drug trials that have been done in aphasia. They are few and far between. I would recommend for any student out there this review chapter by Yano and Small. This is in the Neurobiology of Language book, and it's called Pharmacotherapy for Aphasia, and it's an excellent overview of what has been done in previous studies. All I would like to say about the pharmacotherapies for aphasia so far is that they have been, at best, shown very modest improvement, and at worst, absolutely no benefit at all. Another area that I think has garnered a lot of attention is electrical brain stimulation as something that can be done along with aphasia therapy to try to improve aphasia therapy outcome. Now, both in the areas of transcranial magnetic stimulation or TMS, or low current transcranial stimulation, either transcranial direct current stimulation or transcranial alternating current stimulation. Unfortunately, the studies are very few and far between. Equally as unfortunate, I think that there have been far too many review papers so far in this area. If you look at this literature on TDCS, I think there's a lot more, I know for a fact actually, there's a lot more review papers than there are actual empirical studies, and that doesn't make any sense. Here's one that even got published in the Cochrane Review, where the main outcome that they looked at included only 3 studies that they combined to look at recovery. What they found was that there was a statistically significant effect for improvement in naming, but that was not really the focus. What they were looking at was whether transcranial direct current stimulation could improve functional outcome above and beyond the actual aphasia therapy, and they found no effect there. But I would say this paper is way premature, and I would say for anybody who is thinking about a review paper in TDCS to hold back, and maybe they don't need to be slapped around like this dog right there, but he seems like a bit of a jerk, but at least think about it twice before you start taking on another review paper on TDCS and aphasia. The time is not there yet. We need far more empirical studies to be able to say anything about whether this is going to ever become a part of the solution, but the jury is certainly out. So moving forward, I've talked about behavioral therapy, drug therapy, and electrical brain stimulation. I certainly think that behavioral therapy, which is really up my alley and something that I've spent a lot of time on, that is going to be a part of any kind of a solution moving forward. I can't see that curing aphasia will ever leave behavioral therapy behind. I think that we need to keep focused in improving our behavioral therapies. We need to understand what works best for individual clients or patients depending on where in clinical care you are. I think that perhaps drug therapy and electrical brain stimulation might be parts of the solution in the future, but certainly with regards to the second 2 there, I think the jury is very much out, and not saying that I don't think these things will be important, but rather that we need a lot more research to show one way or another whether this helps out or not. So what I'm going to turn to next is something that I think is one area that we might want to explore as a potential for aphasia cure, and that is a brain-machine interface. So if you don't know about brain-machine interfaces, I'm going to explain that here and then show a couple of videos. Brain-machine interface is really a hardware-software interface that can translate neuronal information, so information from the brain, into a signal used to control an external computer or, for example, a robotic arm. This was primarily developed in animal models, and there's been a lot of human phase 1 trials for neurological diseases. And I would say this is a rapidly developing field. Some of the things that are being tried today I would say 10 to 15 years ago seemed like a complete pipe dream. My interest in brain-machine interfaces was really started with this book here by Miguel Nicolelis. For any students or anybody who is interested in brain-machine interfaces, I would say look up his lab. Nicolelis is at Duke University here in the United States and has done a lot of the seminal work in this area, mostly in animal models. And the other inspiration for me was from this book by Georgi Busaki, who I really have taken a lot of, what I've learned a lot about just brain organization and what drives cortical activity for many different aspects of human processing. So a brain-computer interface or a brain-machine interface in its really simplest terms, and I emphasize simple here, you can record some kind of signal from the brain. There's a lot of aspects and computational power that goes into pre-processing. Then you attempt feature extraction. You try to figure out what is the meaningful signal as opposed to the noise. And then you try to classify that signal so that you can use it to control some kind of an interface. And there are many different things that have been tried, and I'm going to show you a couple of those. The first video that I want to show you is from a video from an empirical study by a French group led by Benabid et al. Their study was published in the Lancet Neurology, and they were using an exoskeleton that was controlled by an epidural wireless brain-machine interface in a tetraplegic patient. So let me see if I can actually play this here. See. Hold on. Ooh. There we go. All right, so let me see, let's get back to, so I think this is pretty remarkable given that this individual has bilateral electrodes implanted on the motor cortex, and then through some very extensive training is then able to operate both this exoskeleton for both upper and lower motor movement. You might think that this is very primitive at this point, but I see no reason why progress in this area is not going to move to the point that at some point we are going to have tetraplegic patients who will be able to walk with the aid of an exoskeleton. Maybe more appropriate for our purposes today, I want to show you also another major improvement with brain-machine interfaces. This is from Eddie Cheng's group at UCSF, University of California, San Francisco, here in the United States. And the title of their paper was Speech Synthesis from Neural Decoding of Spoken Sentences. They also have an excellent video that shows how their brain-machine interface works.
Imagine you're paralyzed and can't move or speak. How would you communicate with the world? Assistive devices can enable people with paralysis to type sentences letter by letter at up to 10 words per minute. But that's a far cry from everyday conversations, which take place at about 150 words per minute. New research from UC San Francisco shows it's possible to generate synthesized speech directly from brain signals. Speech is complex. Brain signals precisely coordinate nearly 100 muscles to move the lips, jaw, tongue, and larynx, shaping our breath into sounds that form our words and sentences. In the UCSF study, volunteer participants being treated for epilepsy read sentences aloud while electrodes placed on the surface of their brains measured the resulting signals.
Shipbuilding is a most fascinating process.
A computational model based on that data enabled the researchers to decode how activity patterns in the brain's speech centers contribute to particular movements of the vocal tract. These simulated vocal tract movements were transformed into sounds to generate intelligible, synthesized speech.
Shipbuilding is a most fascinating process.
Compare that to the participant's actual speaking voice.
Shipbuilding is a most fascinating process.
Now the synthesized speech again.
Shipbuilding is a most fascinating process.
The sound patterns of the individual words synthesized from brain activity are remarkably similar to the original spoken sentences.
Shipbuilding is a most fascinating process.
The model can also decode the acoustic differences between participants' voices. Here's synthesized speech decoded from another study participant.
Addition and subtraction are learned skills.
Addition and subtraction are learned skills.
Even when participants mouthed sentences but did not speak them out loud, the model produced an intelligible synthesized sentence.
In this perfect.
These results represent a critical first step towards one day restoring a paralyzed individual's natural rate of communication, and, most importantly, improving their quality of life.
All right, let me see here. So brain-machine interface for aphasia. This may seem a little bit far out, but I'm going to take a step back here for a few slides, and I'll come back to brain-machine interfaces. But this is going to be important when we start talking about what I have in mind. The first thing that I want to highlight, moving on, is that when we talk about any kind of a speech and language skill, if you will, or any kind of anything that we test, for example, on a typical aphasia battery, or with a test in the lab, that the brain really uses a large network for processing. The brain is not modular. It really uses a brain network that is connected, and the connections between those brain areas allow that network to work in harmony to process speech and language. This is from one of our previous papers, just showing that damage, which you see highlighted in the yellow to red colors here, does have an effect on different types of different aspects of speech and language processing, but also it's the connections between those damaged areas that matter just as much. So both localized and distributed activity is very important for speech and language processing. In addition to that, what we highlighted in that paper is that there are certain areas of the brain here at the top that are especially important for language processing. These are the areas that, when damaged, caused the most disruption across the different speech and language tests that were used to administering. And these are the areas here at the bottom that, when damaged, caused the greatest disruption in different networks. These are not surprising to anybody who studies the neurobiology of language. This is primarily the pars opercularis, part of Broca's area here, all of Broca's area, and then posterior portions such as the superior middle temporal gyrus, and then certainly inferior parietal lobe and angular gyrus in the brain. These are the areas that we typically talk about as the classical speech and language areas. It's not just that those areas function, it's not that those areas function by themselves to give rise to speech and language, but rather they work as a part of a larger network. So one possible target, and this comes back to the individual differences that I was talking about before. One possible target for brain-machine interface is to cure aphasia, might be Broca's aphasia. In the chronic phase, this is somewhere between 30 and 40% of chronic cases. And I really like this definition from Goodglass and Kaplan. So they define the spoken language in Broca's aphasia as dominated by reduction or suppression of speech output with relative sparing of auditory comprehension. Speech production is effortful, usually limited to word groupings of 1 to 3 words produced with labored articulation. A grammatism is common, but not a universal feature. So I like this definition because I think that it applies to a subgroup of folks, whether you like the classical aphasia, using their classical aphasia types or not. I would say that they are still important for many different reasons. One of them, which we will show in our studies later this year, that your response to impairment-based either semantic or phonological therapy is aphasia type specific. Also, the brain damage that gives rise to Broca's aphasia very much distinguishes it from other types of aphasia. In one of our previous papers, this was in the journal, I think this was in Cerebral Cortex in 2015, Fridriksson, first author, we showed that, not surprisingly, predicting aphasia type based on damage, the area that really strongly predicts a Broca's aphasia is damage to the pars opercularis, not pars triangularis. So those are the two main subdivisions of Broca's area. Obviously, Broca's area is not a single area that does a single thing. I'm not suggesting that. Broca's area is really subdivided into many smaller subregions. But when pars opercularis, this posterior region of the brain, is damaged, it does predict that the person is going to have Broca's aphasia. However, that's not enough. And this was shown in a seminal study back in the 70s, in 1978, by Moore et al. Broca's area damaged by itself does not cause Broca's aphasia. What we found in our paper is that areas that are further back in the brain, specifically in the posterior superior temporal gyrus, that for participants to have Broca's aphasia, they need to have damage in at least 2 regions. One is posterior portion of Broca's area, and the other area is the posterior superior portion of the left temporal gyrus. In those participants who had damage to both of those regions, and of course, almost nobody has only damage to those regions. Most of the time, the damage goes far beyond those regions. But if it includes both regions, it's highly likely that that person will have Broca's aphasia. One of the things that we have found that is very interesting about Broca's aphasia, the non-fluent speech that you see in Broca's aphasia, the very limited speech rate, can actually be improved when folks with Broca's aphasia speak along with a speech model, an audiovisual speech model. So they speak in unison with a clinician or a videotape model. We call this speech entrainment. Others have called it choral speech. The first time I came across this was in a study by J. Rosenbach et al., where they suggested this as a part of a treatment for apraxia of speech in adults. I actually happen to think that since then, this is not particularly good primarily for apraxia speech, but rather more appropriate for something like non-fluent speech that we see in Broca's aphasia. Incidentally, at the time, I contacted Jay and I asked them where they got the idea for this kind of choral speech as part of their therapy program. And he suggested that many of the clinicians that he had come in contact with in the community were using this, and that's what gave him the idea. I don't know what is the earliest example of this, but I did come across this study right here. This was actually emailed to me by Greg Hickok at UC Irvine, where he came across this study way back in 1914 by Desjardins and Thomas, where they described the restoration of language in Broca's aphasia. And this was a case where they actually did autopsy as well. You can see a picture of the brain in this of this individual that they studied. And what they described was something that they called the visual approach. Now, I can't verify for sure that what they were doing was speech entrainment, but it seemed to be something similar. And I tell all of my students today, if you think you've come up with a new form of aphasia therapy, check the literature. There's a very good chance that somebody else did it before you. So in many ways, I feel like what we keep doing is recycling old ideas, which is why I want all of my students to go back and understand the literature, starting certainly with the days of Broca and then on. So what is speech entrainment? I'm going to show you a couple of videos to understand how this works. Many of you may have seen this in the past. This is an individual with severe Broca's aphasia who's 22 years post-stroke. He's going to be talking here in the first video about how you make scrambled eggs. And that's without the aid of speech entrainment.
Try to say what you can about eggs.
Eggs. Eggs.
I like to
eat eggs.
I like to eat eggs.
And
all right, so that was him trying to speak with the just spontaneous speech. And then here we see him speaking with the aid of speech entrainment. For those of you who haven't seen this, let me explain real quick. What he is watching is the screen that you will see in the lower left corner, and he's listening to the speech of the speech model through headphones, and he is told, to mimic that speech in real time.
Try to say what you can about eggs.
Yeah, it's the same video. Sorry about that. Let me go to the next one. There we go. So first you hear the audio of the speech model, and we slowly fade over to him after that.
I like to eat scrambled eggs for breakfast. I like them
because they are fast and easy to make eggs and knit up in pan and melt some butter over medium heat. I cracked the eggs into the pan and sure. I like scrambled eggs best. No, until they are done.
Okay, so this is speaking with the aid of speech entrainment. What we found is that there's nothing magical about this one individual. We find that in most folks that who have Broca's aphasia, or at least classified as such on the Western Aphasia Battery, it worked for them as well. We've even tried this in the acute care with folks with very non-fluent speech, and it actually does work in the acute care quite well. Here's that individual's brain. You can see very large lesion in the left hemisphere there that destroyed a lot of tissue, including certainly Broca's area is all gone, and parts of the posterior superior temporal gyrus are very much affected. For anybody interested in speech entrainment as a possible therapy program, I always refer them to Speak in Motion. or what they call VAST. This is, I think, Darlene Williamson and her group. If you really think that this might work for somebody that you're helping, I would strongly suggest that you get in touch with Speak in Motion. I have absolutely no affiliation with their group, but we have found that in the past, the folks who have asked us about this have gotten very good services from Speak in Motion. So we have looked quite a bit at speech entrainment in many different studies, and what we found that those individuals that speak better with speech entrainment compared to their spontaneous speech, that is, that you see increase in just the simply in the number of words produced with speech entrainment compared to when somebody tries to describe a picture, that if you run that ratio in a lesion symptom mapping study, so you're just looking at what is the damage in the brain that predicts better speech with speech entrainment compared to speech description, that damage is very much in Broca's area, specifically the posterior portion of Broca's area right here. So this is Broca's area, the anterior and posterior portions, triangularis and opercularis, and almost all the damage that predicts whether somebody speaks well with the aid of speech entrainment is in that same area. Incidentally, you could say, well, this is the same area that predicts whether somebody speaks well who actually has Broca's aphasia. We also looked at who are the folks that benefit from speech entrainment. We compared their output during speech entrainment and picture description. If somebody spoke with very similar output on both, they got a score of 0. If they scored better, if they did better with speech entrainment compared to speech production during picture description, they would score above 0. And if they did worse with speech entrainment, they would score below. And what we found, if you just look at aphasia type, almost all the individuals who had Broca's aphasia were indeed producing more speech with the aid of speech entrainment compared to spontaneous speech. I realize there's a lot here, and I promise I will bring this back around very quickly to brain-machine interfaces. What I think is going on in Broca's aphasia is that this is a motor problem that is caused by an impairment also in what we call an efferents copy. This is a model by Perel and Hood of the speech production system, that you can think about it this way. As we initiate motor movements for speech production, at the same time that we have that feed forward signal that produces that speech, we also need feedback signal from posterior regions, posterior parts of the brain that monitor that speech. That template that is used to monitor your speech output is what is called an efferents copy. And this comes from motor literature that was not speech specific, but it's very clear that any skilled motor movement relies on an efferents copy, so that the brain knows, here's the output, and once I execute that output, here is what I should feel, which is one of the reasons why we can correct speech very fast on the fly once we make speech errors. What I think the case with Broca's aphasia is that that efferents copy, or that feedback, is not working correctly. The reason why speech entrainment works is because it essentially provides an external efferents copy that you can then entrain your speech to. So you're not having to rely on feedback from posterior regions. Once you have that external feedback, you can then produce fluent speech. I have given lectures on this in the past, and I plan on doing some more, explaining better my thoughts on speech entrainment and why it works well in Broca's aphasia, and in continuation of that, why I really think that Broca's aphasia is essentially an efferents copy problem. And I hope to do that on the C-STAR platform sometime soon. One of the things that we have found out with speech entrainment in a recent study by Lisa Johnson, Lisa is a Ph.D. student in my lab, was that we looked at functional brain connectivity between two different regions in the left hemisphere that we think are very important for speaking with speech entrainment. The first one is this posterior portion of Broca's area, or pars opercularis. And based on some other research that we've done, particularly two studies, one with Leo Bonilla at MUSC and one with Greg Hickok at UC Irvine, showing that the posterior middle temporal gyrus of the left hemisphere is very important for being able to speak with the speech entrainment. In the participants in this study, there were 24 of them who had Broca's aphasia, and we also had 20 control subjects. We had them all attempt a spontaneous speech task while they were undergoing MRI scanning, and they also did a speech entrainment task. And we simply looked at what does their functional brain connectivity, that is, what is the synchrony like between those different areas when somebody tries to speak or produce spontaneous speech, as opposed to when somebody speaks with a speech entrainment. And that's where we get to this graph right here. We normed everybody's functional connectivity or coherence between the different areas of the brain, both in the left hemisphere and in the right hemisphere. And then we looked at the coherence depending on whether they were doing the free speech task, which is what the FS stands for right here, and whether they were doing the speech entrainment task, which is what SE stands for. And then we simply compared that across all these different connections, and the connections are color-coded. So here's the connection between the left hemisphere, pars opercularis, and posterior middle temporal gyrus. Here's the connection between the left posterior middle temporal gyrus and posterior right temporal gyrus, and so on. So you can see that this is color-coded. There were only two areas of the brain where we saw an increase in connectivity when the person spoke with the aid of speech entrainment. The first one was, not surprisingly, between the anterior and posterior speech regions in the left hemisphere. We also saw a improvement in the connectivity between the posterior left hemisphere and the right pars opercularis. So those were the only two connections that we see an improvement in connectivity, which we think is important for inducing fluent speech. So all this is to say, I'm going to bring it now back to, let me see. Yeah, I'm going to bring this back to speech entrainment and why we think that it works in Broca's aphasia. So speech entrainment, we think, synchronizes anterior and posterior activity during speech. We think that this synchronicity is impaired in Broca's aphasia, which is a part of why this efference copy is not working out. One possible mechanism that we think about then for impaired speech in Broca's aphasia is this lack of synchronicity. So this might be a potential target for treatment. So it could be the case that what we could do is to try to synchronize anterior and posterior activity in the brains of folks with Broca's aphasia, and therefore with aphasia therapy, with the help of aphasia therapy, we could at least partially and maybe fully restore fluent speech. Now, so how might this work? A very rudimentary process, and I understand that I'm probably going to start running out of time here soon. We would have to do the same thing that the other two groups that whose research I showed you earlier. This would have to happen with some electrode implantation, both in anterior and posterior regions. I think that this would have to be functional MRI guided. We would record the signal in both cases from the anterior and posterior regions. It would go through the same processes as most brain-machine interfaces go through. That is pre-processing, feature extraction, that is what is the signal that matters, classification of that signal into something understandable, and then try to synchronize that activity. I don't think that this is going to work simply by temporally synchronizing those regions. I think that it's probably going to be a lot more complicated than that. We need to understand what the posterior regions are doing, but understanding that synchronizing them in some way, whatever that synchrony might mean, needs to be so that it allows crosstalk between anterior and posterior regions. I put the aphasia therapy here on top because I think at some point we need to figure out whether where in the process the aphasia therapy is going to help out. So I think the first thing to do before, obviously, we're not about to implant anybody with electrodes anytime soon. But I think the first, maybe one first step for proof of concept would be to use what we call transcranial alternating current stimulation to try to synchronize anterior-posterior activity in the brain. There's a recent paper that came out by Reinhardt and Wen in Nature Neuroscience. This was a very important paper in that they used transcranial alternating current stimulation. It's a method that you can use to actually manipulate the timing of neural activity in the brain. And what they found was that by targeting the hippocampus in older individuals, they were able to restore short-term memory in those individuals so that it looked exactly like what you would see in younger individuals. So you can do that by using transcranial alternating current stimulation. Using a similar approach, I think then what we would need to try is whether we can identify the residual brain areas that are still involved in language processing but might be disconnecting, and as importantly, we're involved in some kind of a process that is related to what the damaged brain areas were doing before, that those areas can be retrained, and then by connecting those areas, we'll be able to restore speech production. We have a project that is underway looking at exactly this. This is led by Dr. Laura Lee Johnson, and also two of my Ph.D. students, Lisa Johnson and Lindsay Cater, where we are looking at whether using TACS will indeed at least minimally affect speech output in real time in persons with Broca's aphasia. I think that if this actually works, I think that then it will open up a whole host of things that we could consider as a way to restore speech fluency in people with aphasia, using then hopefully sometime in the future a brain-machine interface, literally implanting the cortex on somebody to try to resynchronize connectivity. So I know this may seem like a pie in the sky. I don't think it is. Maybe my theory is wrong, and maybe that's a pie in the sky, but the actual implantation is not. So if you know anything about the company Neuralink that is now underway by Elon Musk, what he has been working on is an actual brain-machine interface for folks without brain damage, simply banking on using a brain-machine interface that then individuals can use to manipulate the outside world. So the way that they think about this is that instead of using your hands or your fingers to operate your phone, you will simply have to think about what you want to do, and it will operate your phone. Same thing could be said about, flying an airplane, driving a car, manipulating the electricity in your house. They are literally working on this. They have a prototype working, and they are starting with patients, but the idea is to do it so that this will be not just usable among the general public, but this will be accepted by the general public. Otherwise, I don't see how their business model is going to work. But I would say this will happen. I think it's highly likely. There's a lot of work that needs to be done to make sure that all the kinks will be worked out. But I certainly don't see it as a pie in the sky right now. At least the concept of implanting the brain to manipulating the outside world. And in our case, I'm not describing a traditional brain-machine interface. What I'm really talking about is a brain area to a brain area interface, rather than something that manipulates the outside world. So I'm on the home stretch here, curing aphasia. So what is a reasonable timeline for a project like this? I think in the next 5 to 10 years, neural implants are going to become safer. This will make trial and error certainly a lot more feasible. I think that the research that needs to be done, certainly starting with TACS, some of those studies hopefully will come out in the next 5 to 10 years. If that proves that that works, I think that then moving on to the brain-machine interfaces is straightforward. The signal processing part, certainly there's a lot that needs to be worked out, but many of the algorithms that are already being used have been developed. There are obviously projects that have already used very extensive computing power to try to figure out how do you translate the signal from the brain into something meaningful. What we need to do is to then translate between anterior and posterior activity, and I would imagine that artificial intelligence is going to play a huge role in figuring out how that works. So the first prototype brain-machine interface or brain-to-brain interface for aphasia treatment, if things work out like I think it's possible, I think we might see that something like that in 10 to 20 years. I completely understand that I might be wrong here, and this might be a shot in the dark, but I see this as one area that we not only can, but probably should look into as a way to help people with very severely limited speech output to not only improve their output, but hopefully get them close to something that is premorbid status. So the last couple of slides that I want to talk about specifically to students. I hope, like I said, I hope there are some students on there. Those are the folks who are going to be taking over this field or are ultimately going to do the most important research in this area and just in curing aphasia in general. I just want to talk a little bit about my experience. So to anybody who is a new faculty member or a student in our field right now, obviously, and you've heard this before, I would emphasize that the path ahead is certainly complicated. This is not simple stuff. It's going to be difficult, often discouraging with lots of setbacks. But I've also found that this is a very rewarding field. It's exciting, and most importantly, it is necessary. We need people who are a part of the solution in improving the lives of people with aphasia, not just with regards to something that I'm describing right now, but I think we need to get a lot more folks interested in curing and helping people with aphasia, and that we need more targeted research than what we've had in the past. So what does it take? I think that for students, you need to get good training. There's nothing that comes in place of that. Obviously, you have to have an interest in helping others. You have to have interest in both science and the clinic. I think that if your interest is primarily clinical, find somebody who has been working in neurophysiology or neuroscience that you might be able to team up with. Or if you're more in the neuroscience field, make sure that you talk to the clinicians. It is very important that you understand what the real questions are that are going to move the needle, that are really going to make improvements in the field. Without talking to the clinicians, I think that there's a chance that you will miss what is very important. And then last, but certainly not least, you really need to write grant applications. To get large projects, certainly anything that I talked about here today will not work on a shoestring budget. This is expensive. Writing grants, I understand, can be very arduous. It is often very discouraging, but I would say keep at it. If you don't give up, chances are that you will be successful, and that you will then, one of the ways that you will do help out is, but one of the things that is going to help you help others is to do these large projects that are so important. My last slide here, I want to thank everybody who I've worked with. I started putting in the pictures of all the different people who worked with me in the past and are working with me right now. It is a huge group, and I'm only one very small piece of that group. I've been very fortunate to work with some excellent scientists, not only here at the University of South Carolina, but also at different institutions here in the United States, and even had a lot of crosstalk with my colleagues now in Australia. That's something that I'm hoping that we can capitalize on and even improve further in the future. And last, but most importantly by far, I want to thank all the people with aphasia who have participated in our research in the past. Not only are their data extremely important, but it's been very encouraging to me to see some of those people persevere through great difficulty. And whenever I feel sorry for myself with regards to maybe lack of progress, or another paper wasn't accepted, or another grant wasn't funded, I think that going back and spending time with folks with aphasia will help you feel re-energized and give you what you need to move forward. So I'm going to end at that. Thank you, everybody. And I think from Todd, we'll be able to take some questions.
Can you hear me, Julius?
Yeah, can you hear me, Todd?
Miracles happen, sir.
All right. Excellent.
We do have a couple of questions. The first one, if collaboration across approaches is important, and most believe it is, wouldn't it be useful to look at the impact of neurobiological approaches beyond naming to see the impact on daily life participation and conversation?
Yes, that is very important. So we are actually working on a project like that. So Lisa Johnson, who is a PhD student with me right now, is looking at exactly that. So instead of, understand that naming, I understand that it's been criticized a lot, but naming is a proxy for something. Naming, your impairment in naming is strongly associated with your aphasia severity. Absolutely no doubt about it. All the data show it. And if you can improve lexical retrieval, at least theoretically, if you can lessen the language impairment, it should translate into functional outcome. Now, that has been criticized a lot in the past. I think the main criticism comes from this problem that I talked about earlier. Most of our studies are small. What we find is that a lot of participants don't improve, but that does not change the fact that many participants that improve lexical retrieval, for example, show improvement in discourse. Now, some of the research on that is going to be coming out from my lab in the next year, and I know there are other people looking at this, but you're right. We ultimately want to look at not just something associated with a given process that may not seem ecologically valid. We want to make sure that we're also looking at ecologically valid stimuli. And like I said before, we are working on that. I would say watch that space. I'm hoping that later this year or early next year, our first paper will come out on that. And if you want to write it down, Lisa Johnson is going to be the first author on that paper. I really think that it's going to be a different way of showing us that how our brains work in literal conversation or listening to others talk to us, how that synchrony occurs and how we synchronize with other people. So that when we talk about communication at the neural level, as you're listening to me right now, you can think about it as you and I are entraining our brains. We are synchronizing our brains. So a lot of the activity in my brain that goes into producing speech right now literally is recreating exactly that same kind of neural activation in your brain. So that neural synchrony, I think, is crucial for effective communication. And like I said, we are going to be publishing something on that soon.
Wonderful. If other folks have questions, you can certainly use the chat feature to get to us. Another question, Julius, could you say a bit more about the mechanism underlying the synchrony therapy? The example from Chen showed simulation of a specific measure. Are you envisioning a more generic coactivation as the mechanism?
Yes. So I think synchrony by itself is going to be important, but I think that ultimately, until we start looking at the differences in the anterior and posterior features, but essentially the features that matter, we don't know whether this is just going to be some kind of a one-to-one synchrony between activation in those areas. I think that would be a good start, but translating what posterior regions and anterior regions are doing, in both cases being very important for speech production, but how that translation happens, I think there's a lot more research that needs to be done on that.
Okay. Another question. With such variability of both functioning and response to treatment by people living with aphasia, are large randomized control trials truly feasible and the gold standard for aphasia treatment research?
Yeah, so okay, that's an excellent question. I literally could talk for another 2 hours on that. Randomized controlled trials, first of all, demonstrate to people outside of our field who don't take anything as being legit evidence, except for randomized controlled trials, it sells to other people that are consumers of our research and who we rely on that we need to justify our therapy to, they understand that phase 3 clinical trials are important. Now, it might be the case, and what I suspect is that if we, instead of looking across all participants with aphasia, perhaps a randomized controlled trial could focus on a subgroup of people with aphasia. And what I suspect is that what we find, certainly from our trial right now, where we enrolled over 100 participants, now looking at the baseline factors that predict outcome, simply using those factors to stratify a new clinical trial, I would bet a pound to a penny that I could show an improvement in all the participants. If I was able to use baseline information to select who is and who is not included in that trial, I think we could do a lot better. Now, I understand that some people might think that that's not feasible because you would exclude a large group of people. What I would say is maybe the folks that are being, well, not maybe, but I do think that the folks who are excluded from that trial, maybe they will respond with another therapy approach. So therefore, they could be randomized into a different study. I'm not saying that we should look at aphasia, and I hope that this came through here. Individual differences are important, but they are important, but they certainly do in no way cancel out the importance of phase 3 trials.
Okay, we've got time for 2 more. How do you account for word finding deficits in Broca's when evaluating the effects of entrainment, which gives them the words? Does spontaneous speech improve after treatment with I'm missing the type word, but hopefully that's making some sense to you, Julius.
Yeah, so what we have shown, certainly in our first study from 2012, this was Fridriksson et al. In brain, we did show that speech entrainment therapy, this was 6 weeks, does improve spontaneous speech production. And when I say spontaneous speech production, I want to say there's some caveat here. We're talking about picture description. So what we have shown since Helga Thors, who was one of my PhD students who recently completed her PhD, we are going to be publishing these data as well, in 20 participants with aphasia receiving a speech entrainment therapy, literally speaking with the speech entrainment over a 3-week period, so 15 sessions, or maybe it was more, maybe it was 20 sessions, we see an improvement not only in things like speech in picture description, but also on other discourse activities.
One last question, and it kind of goes to the heart of the series, which is why I saved it for last. This person noted their curiosity in why the series is labeled as a cure for aphasia, and wondering if you're concerned about overpromising the outcome of these methods to people with aphasia.
Yeah, this is not a talk for people with aphasia. It just isn't. It is a talk for the people who do the aphasia research. And like I said at the beginning, this sounds weird to be talking about cure for aphasia, but I think that it's healthy to say we need to be looking towards a cure, because the way that our field has been going forward for the last 40 years, I can imagine that many people with aphasia, if they understood where we were 40 years ago and where we are today, that they would be satisfied with that progress. I certainly am not. And I do think that we need bold ideas. Maybe some of those ideas are never going to work out, but we'll never find out unless we try it.
Wonderful. Well, Julius, thank you. I would have thanked you up front, but the technology just didn't want to play with me very nicely today. But I'm more happy that the tech and the slides and the audio was coming in just fine from South Carolina, which was easily the more important part of this process. Appreciate you allowing y'all having Aphasia Access involved with this. We're honored to be a part of the series, and a sincere thanks to everyone who has been on the line for the last hour and a half, including getting through the first couple of minutes with our audio. We will take this presentation and we will get it up online as soon as we're able. Our goal is generally within a week, hopefully a little bit faster, and we'll let everyone who registered know first once it's available. So thank you, Julius. Thanks, everyone.
Todd, can I say something really quick? Todd, can I just add one more thing real quick?
Please, of course.
Yeah, I want to emphasize, I don't want this discussion about what I talked about. I don't think it's a good idea to do it on Twitter or Facebook. I tend to think that those conversations always leave something to be desired. I never think they really get to the bottom of anything. And as one of the questions indicated, I think they could give the wrong idea to people with aphasia with regards to what I'm talking about, so that they think, oh, they're going to be curing aphasia in the next 5 years. Obviously, if you look back at my slides, that's not what I think. And I said it multiple times at the beginning, this may never work out, but that's the way that I see one avenue for treatment. So what I would suggest, find my email, shoot me an email. I would love to have a conversation and get feedback from you. But, you know, I realize that some of you might think that this is complete nonsense. I would appreciate it if you didn't post it on my Twitter feed.
Of course.
All right, thanks.
All right, all. Thank you so much, and enjoy the rest of your Tuesday.
All right, bye, Todd.
Bye.